Now, what if there's not? What if that doesn't work? So if you have a membrane, a semi-permeable membrane, and if it helps you to think about a phospholipid membrane, do that. But otherwise, just think of a membrane that allows some things to go through and others not. And it usually lets water go through. In this experiment, this is really pretty impressive for a visual, actually. So you start out with both solutions. So you fill some water into a U-shaped tube. And obviously it's going to level out right at the same side on both sides, which in itself is kind of impressive in and of itself. And if this is clean water, this is just going to be that way. However, if you're going to do this on the right-hand side, you have, for example, a sugar solution. And on the left-hand side, you have a just plain water or a less concentrated sugar solution where you have fewer solute molecules over on the left than you do on the right. Then what's going to happen is, I'm going to clear the pointer here real quick. What's going to happen is these guys want to go over there. They would love to, right? The little purple ones here, they would love to go over to the other side in order to equilibrate this, but they can't because it's blocked, right? So instead what happens, this force of entropy is so strong that the water, in fact, moves over to the point that water actually goes against gravity. It actually goes up to some level. Obviously, gravity is working against it, right? But the water tries to equilibrate it. One of the ways I find for students to think of it, when you think about when you're thinking about what the water does, is think about the water diluting something. So the direction of the water is always that the water dilutes the solution. So whatever direction the dilution goes, that's where the water goes. So, and we're going to talk about that in just 1 second when we talk about the osmosis in cells. Now let's spend a minute here talking about this. Thinking through this, you first have to know that tonicity is a relative measurement. Is something is isotonic compared to something else. So it's not an absolute measurement, it's a relative measurement. So an isotonic solution, the solution and the cell have the same solute concentrations. So water goes in and out. It's not pushed in any direction. It's not compelled to go in any way, one way or the other. In the case of an animal cell, when an animal cell gets into a hypertonic solution, now think in terms of dilution. So what happens is there's more hypertonic, meaning it means there are more solutes out here in the area out here than there are inside of the cell. So there's more crud on the outside than there is inside of the cell. So if water wants to dilute everything, what direction is the water going to go? Obviously, the water's going to dilute. It's going to move out. And that is actually not a problem for an animal cell. On the other hand, if you put an animal cell in a situation where it's in plain water, there's more stuff on the inside of the cell than there is on the outside. This is called hypotonic solution. Hypotonic solution. You have lower solute levels on the outside. And then when the water tries to dilute things, the water goes inside of the cell. Now with plant cells, the situation is slightly different. It's the same idea with the isotonic, right? But plants, you guys know plants do okay in regular water. Well, that's because they have a structure that animal cells don't have. That structure is the cell wall. And the cell wall is a rigid structure that keeps plants standing upright. And so in their case, it's actually a good thing that there's this turgid pressure that comes on the inside where the water is pushing in and it's pushing against that. It's filling the vacuole. It's going into the cell, which is exactly what the plant wants, right? Because it wants the water to come inside of the cell. So in hypertonic solutions, plants are extremely happy. Plants are extremely unhappy in a situation where they're like in salt water, because what happens there, the same thing as with an animal cell, is they shrivel on the inside because the water tries to dilute the outside. Um, but they have this plasma membrane that's attached to the cell wall, and then what you see there is you get plasmolysis, which can be the death of the cell. And that's hypotonic, hypertonic, isotonic for you. That's osmosis for you. Now let's give you a real life example. A little paramecium over here is a single cellular organism that just lives in fresh water. It's got a little contractile vacuole. What do you think that vacuole is doing? Is it making water go out, or is it making water go in? Is he drinking, or is it purging? What do you think? The water is going into the cell because there's more crud here, right, than there's on the outside. So this little vacuole is going to be expelling water the entire time. And if we give a drug to this poor little paramecium that paralyzes this vacuole, it'll blow up. It's very sad.